Audio check, 1, 2, 3.
Is this the other one?
Yep. And go ahead. Fill in the screen with that, with the, oh, you got right to the middle. I'll hand one to you. Surprise you. That's a line from Ratatouille. Yeah. Anton Ego. Surprised me. Thanks everybody for coming out tonight. It's a beautiful night. Next week, Flag Day, June 14th. Beyond the Tailpipe, the key to driving green. Kelly Senecal from Engineering Interdisciplinary Professional Programs will be here. He does computational fluid dynamics. Yeah. I'm just going to do speed abacus and see if I can keep up. And then in 2 weeks, we have a special Wednesday night at the lab, which will be at 7 o'clock, but it won't be here. It'll be across the street in 420 Henry Mall. I made a mistake on the missive today and said 425, even though I got the name of the building correct. The room is 1125. It's up on the second floor. That's why it's called 1125. So if you want to come in the front door, bring your mountain climbing gear, because it's very steep steps from like 1915 or 16. Plan B is to go around to the southwest entrance of the building, come in on the ground floor. You can roll and stroll without having to take a step into the elevator. In the elevator, hit the button for 1, I hope. It'll take you up to the 1100 floor and then roll and stroll down to 1125. Say again? You know, I could call them up, but I don't think they do it for me because I don't work in that building, so I'll have to ask somebody else. But the PBS Wisconsin crew will be there at 5, so I'd be able to call somebody and we can try that tape of duck again. On June 28th, Jim Lattis is going to be here because it's the week of the annual, although there was a hiatus for COVID, meteorological high school summer camp. That's 4 days and 3 nights here at Madison. And so that's one of the things we've been doing for many years, is having a speaker for that group. Jim is he's an astronomer, and he's going to talk about preparing for the full eclipse of the sun. Oh, wait a minute. Total eclipse of the sun in August of 2024, which is going to go through pretty close to here. And I missed the one in
It's going to be in April 8th.
The doors will not be opened. So show up with me in August and down in central Illinois, and we'll have a great time. What's that?
Are you buying? We're going to have lots of extra time interviews.
Oh, that's true. Well, I would not make it as a Connecticut Yankee in King Arthur's Court because I'd be 5 months late. Anyway, so thank you very much for all that good stuff coming on. Tonight, it's my pleasure to introduce to you Laura Hernandez, Alysia Vang, and Ariana Negriero. And they're all from the Department of Animal and Dairy Sciences. And Laura Hernandez is a professor in that department. Alysia and Ariana are grad students. They're going to talk with us about how data scientists and lactation biologists develop new ways to predict lactation potential in mammals. I'm going to do this in reverse order. I'm going to ask Ariana the questions. If you can give her the microphone, that would be great. And then if you could come over here, because the camera doesn't swing that wide, that'd be, oh look, you're tethered. Ariana, where were you born?
I was born in Dallas, Texas.
And where'd you go to high school?
I went to high school in Fort Lauderdale, Florida, just north of Miami.
And where'd you go for undergrad, and what did you study?
I went to Michigan State University for my undergrad, and I studied animal and dairy sciences.
And then when did you come here?
I moved here just over 2 years ago.
Good timing. Yes. And so you're a grad student now in your 3rd year or so?
Mm-hmm.
Excellent. Yep. Thank you. Now I'm going to ask the same questions of Alysia. Alysia, where were you born?
I was born in Green Bay, Wisconsin.
Is that a who? You have fans. Where'd you go to high school?
Preble High School in Green Bay.
Oh, way to go. And then where'd you go for your undergrad, and what
I
did you
actually came
study?
here for undergrad, and I studied genetics and genomics, and then I got certificates in global health and education services.
She's an overachiever.
And she's from Green Bay. And then when did you start graduate school?
I started graduate school in 2019.
Oh, so you've already got 4 years in. Way to go. And now I get to do and ask Laura. All right, Laura, where were you born?
El Paso, Texas.
And where'd you go to high school?
El Paso, Texas. Eastwood High School, home of the troopers.
Whoa, nice.
Yeah.
Is it named for Clint?
No.
Okay. And then where'd you go for your undergrad, and what'd you study?
I went to New Mexico State University, and I studied animal sciences.
Is that the land-grant university in New Mexico?
It
Is that
is.
Las Cruces?
Las Cruces.
Wow. And then where'd you go for your master's and PhD?
Master's at New Mexico State, PhD at the University of Arizona. Go Wildcats. Not to be confused with the Sun Devils. Or Scum Devils, as we like to call them.
It's a family program, so
It's a thing.
yeah,
It's a
good.
thing.
And then, did you do any postdocs before coming here?
I did at the University of Cincinnati at the med school.
Okay. And do you have anything to say about the Bearcats or anything?
They have a good basketball team.
Ding, 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 ding.
And I liked living in Cincinnati. Lots of sports.
Yeah,
And
yeah.
I love sports. So,
Excellent.
Yeah.
And then, when did you come here?
Oh, wow. June of 2011.
All right.
I keep track by the birth of my daughter.
That's a good plan.
So
they make excellent calendars.
They do.
I'm very grateful that the three of you are here. I'm looking forward to hearing your talk. Would you please join me in welcoming all three to Wednesday Night at the Lab?
Well, thank you. I did this once a few years ago now with a couple of other grad students. While both of them were in my lab, they kind of were sort of given a choice. Ariana very graciously agreed to partake in this, as it is a collaborative effort on a project I've been doing with Joao Dorea in our department as well, who is a nutritionist converted into a data scientist now. He says nutrition is no longer a thing for him, but he's lying. And so it's a project that I'm really excited to talk to you about. And so Alysia works in my lab and Ariana works in Joao's lab. And it really kind of shows you how biology can come together with all of this new artificial intelligence type technology, and how we can really leverage our strengths to develop new ways to study things. And so when Joao started, I think he started in like the summer of 2019. He might have started just before Alysia started graduate school. I have had this passion project. I'm a true lactation biologist by training, and so I study rodents, humans, cows. I really wish platypuses, but my department chair keeps shutting me down on that one. But there is this group of women who go largely undiagnosed, and they kind of get forgotten about, and they have a phenomenon called insufficient glandular tissue. Now, it seems that most people think that women magically get pregnant and then magically they breastfeed their children and there's no problems. Wham, bam, thank you, ma'am. Get the good stuff and move on to your healthy life, and this is a really easy thing. It is not. For any of you who have breastfed, you might know that it is not an easy thing. So right now, the estimate is about 5 to 15% of women are thought to have this phenomenon called insufficient glandular tissue. However, colleagues of mine that are in clinical practice in medicine suspect that this number is probably a bit higher because it's hard to diagnose. Women give up in the hospital, and so no one really actually figures out that it was a problem. They just stopped breastfeeding because something happened. But this is a pretty classical clinical presentation in which the tissue is very underdeveloped. And so this is a way that like people can see as they're being checked in their obstetrics appointments, where you could see that potentially the woman was not going to have enough lactating tissue to support their offspring. And the breasts come in and they look pendulous and they point outwards. And so this is really a typical presentation. No one really knows if this is a genetic condition, if this is a preeclampsia condition, if this is an obesity condition, or it's just something that happens on accident. And there is not really any research on this in women who have taken the diligence to go and take blood samples and really try to identify what's happening within the tissues. So I went on sabbatical now several years ago, and I was working at American Family Children's Hospital and also in the clinic at UW Health. And I was really surprised how much higher of an incidence this is. And we also don't have a lot of preventative treatment, or at least identifying treatment for women that might tell them, I'm not going to make enough milk. And so what happens is they give birth, they have a baby, the baby comes. We already don't produce a lot of milk to start with. Unfortunately for humans, you would wish they'd be more like cows and they would start producing a bit more milk before they give birth, but they actually don't start to produce milk until after they give birth. And so the colostrum amounts are very tiny. And this is a very stressful situation. Women are complicated, humans are complicated, and all the psychological aspects are involved in this. And so a woman then decides, I'm not feeding my child, they're going to die. OB-GYN's there, pediatrics is there, the nurse is there, and everyone's freaking out, and she says, I quit, before she even knows if she actually has reached a potential or has a potential. So there's no predictive measures out there. If you decide that you want to have a lactation plan, they don't really have lactation plans like they do with the birth plan, where you can walk in and say, I'd like a bathtub and music and my drugs at this time, and I want to walk around. Like, they don't even discuss this aspect of it. And this is the long-term aspect, right, of taking the baby home, and what do you do then? Which is what I wanted to know, but no one ever told me. It's good, she's almost 12, she's still alive. So I have had a real passion for this and trying to figure out what to do about this. So I had seen there's a group in Australia that was doing a little bit with using ultrasound. A lot of things in medicine now are being used as point of care ultrasound, so they can come in to the ER and they scan you. Oh, yep, something's wrong with your heart. Yep, something's wrong with your foot. Yep, yeah, here, let's ultrasound. And it's something that's very easily used if you can train people to do this. And I thought, like, this is brilliant. They've done some of this, but not point of care. Maybe this is a way we could think about or reinvent. Let's have a lactation plan. We could do the scans at maybe the 20-week appointment when you go in to have your reveal the sex appointment, if you so choose to do that, and the 32-week appointment where potentially you're gonna have your glucose tolerance test. So we're not adding extra appointments or extra burdens, but it's something that the woman is already typically doing in her normal course of pregnancy. And so I thought this might be a way to kind of go about this. And so could we use this as a way to identify how much a mom might be able to make in milk? Like, maybe we can say, she's gonna be like based on our metrics here, we think she's gonna be at about 50% sufficiency. She can go into the lactation knowing, you know, I'm only gonna make about 50%, so if I have to supplement with formula, I'm not failing as a mom. Or I can choose not to breastfeed at all, or I can say, like, I'm gonna make what I can and supplement what I need to, and she feels a lot better. So about this time, Joao had started, and he's doing imaging of all kinds of things, which Ariana will talk about in a little bit, but he came down to my office and wanted to write a USDA grant, and he had looked at some stuff in goats and in dairy cows using ultrasound as well. And I showed him some of these papers, and I was like, Joao, I think we can do this. Like, let's do a project. This is good for understanding lactation. He was very interested in how he can roll some of this methodology into having a better prediction, not only for lactation in dairy animals, but also maybe to build this into the residual feed intake metrics that they have been doing for a long time, and while good, are not perfect. And if this was another thing that we could add into the calculations, you would have a better idea before an animal is even maybe inseminated, whether you should inseminate her or not, if you want to keep her in the herd, what her potential might be. So he was really excited about this, and unfortunately I can brag about him hopelessly here because he's not here, he decided to write this grant, and the very first one he wrote, not my experience, it got funded. And he's kind of been on this like upward trajectory, and he said he's waiting for the bottom to drop out. But he's doing a lot of really cool things, not just this. So what we decided was that I wanted to use this ultrasound technique. Some of the stuff Ariana is doing is she's doing 3D imaging and mapping of the udder. We're also using the genetic components of our animals because all of the animals in our herd here are genotyped, so we know what the genomic components are. And we're trying to understand and put these pieces together to take genetics, to take AI, to take data science, to take physiology, and have a better understanding of the system so that we can use this kind of long term. And what's really interesting about this is that no one was doing it. So we have these ultrasounds that we go and we're scanning cows with. They use it for a lot of things like detecting bovine respiratory disease in calves. So it was a really a cheap way to maybe sort of look at this. And so because we work with animals and the system is controlled, we were able to take calves from birth all the way through almost their entire lactation, which I'll let Alysia hit on a bit more, this project that she's been working on. So we've been able to have a really beautiful data set where we enroll the animals when they're born, and we have repeated measures across time through their entire life cycle. And we're building the model between taking histology samples of the udder, ultrasound images of the udder, and then we'll have their lactation data and a bunch of other data about their kind of lifestyle, so to speak, so that we can determine how good our model is for the ultrasound at least. And then there's several other aspects of this project. So we have been doing this now since the summer of 2020 is when this project started. So Alysia has this really nice diagram she put together for some presentations she's done. And what we're trying to make use of is that, like I said, we can have this birth stage where the udder is born. When you're born, you don't see much change in mammary gland tissue until an animal starts cycling. So when a female starts to have some level of cyclicity, that's when you really start to see some divergence between males and females in the growth of the tissue, and that's because of the estrogen and progesterone and the effects on like the structures that eventually become the secretory tissue. And so you don't see that until probably 12 to 14 months of age, and then there's a period at which they become pregnant, and we're able to follow them over the course of time so we can look at what the mammary glands look like as they're growing, when they start cycling, when they get pregnant, through their entire pregnancy, through an entire lactation. And Alysia has told me in the last couple weeks she'd like to follow them through a second lactation. So maybe we can get to that point. But taking this life cycle and having these repeated measures tells us, like, instead of having a bunch of animals at the same time, you hope that that data is reflective, but you also don't know what the environmental influences are that you might lose if you're not following that animal over their lifetime. And so that's the real advantage of this model. Plus, clearly we control our diets to a great extent that you don't see in humans. And we also have like control of a lot of different things that you wouldn't in a human population. And we can intake more sampling. So the previous work, like I said, that they've used ultrasound for has been done in goats, and a lot of dairy goats actually in Brazil, and then also in some cows, but largely goats, where what they're looking at is they take an ultrasound image, you can see there on the far left in that picture, and ruminants have a cistern area that is not present in rodents or humans, and that's like what that outer outlining area where the black space is, and that's where you have milk pool close to the teat end of a goat or a sheep or a cow. It's not the secretory tissue, it's just kind of like, I liken the tubes, they're like PVC pipes that drain the secretory tissue into a big PVC bowl that holds it there until you pull the milk out. And so there is some data from quite some time ago that talks about the size of the cistern relative to the amount of secretory tissue and how much that plays into milk yield. And so there has been some really like early kind of primitive sort of things done, but we thought we could take it a step further. And so one of the things that Ariana has been working on is like the relationship between the internal features and the external features of the mammary glands. So that's one of the things that she's worked on. And so that doesn't necessarily apply to Alysia's project, but he and Joao and Ariana have been working on that, as well as some other people in their lab, and then how we can develop these models. And these models are great, and data science is great, but it takes a physiologist and a lot of time and a lot of annotations to build these models. So like a computer gives you a beautiful model, but that model is built off of something that was collected, that has been annotated, so you can train the artificial intelligence in the computer that this is what that actually is. And so that takes in a tremendous amount of time. And Alysia will show you some of that in a second. So kind of our experimental approach in these projects are that we take an ultrasound image, and so we try to take it within the same area. And this, of course, changes as an animal grows, right? It goes from small to really big. And so that's been one thing that we've encountered that's been challenging, like how do we try to get the same area every time? And then you have to take that and input it into the computer program and segment it and process the image using very complex things that I don't understand. If you have questions, thank goodness Ariana and Alysia are here for those types of things, but they use a bunch of different computer programs depending on what they're doing to do that. And then they try to produce these measurements. And so that's one thing we're doing with the ultrasound. Ariana's been doing a lot of work with the 3D imaging, and this is where they take these image collections, and you'll see some videos of her doing that, and they extract the features from that, and they utilize those features and then try to train the model to do this. One of the cool projects she's not talking about here today, but that she's been working on is looking at the mouth movement of the cows and how they sort through feed in a space, because they do sort through feed, and using the 3D imaging you can kind of figure out why they're doing one pattern versus the other. It's really cool. When he first approached me, he's like, you know how they can see your face with the iPhone and it opens? He's like, that's what I'm doing. And I was like, oh, this is way beyond the physiologist in me. So they did some pilot studies at our farm here, looking at some of these features that are not related to our calf project, and looking at somatic cell count. So could you extract some of the features of the tissue and say that this cow would have high or low somatic cell count just by taking an image that would give you like ideas in advance of actually potentially an animal being sick. So there's a lot of different things we can do with these features. So I'm going to let you talk about the feature extraction because you do some of this. You two do this more than I do. They're like nervous to talk about it. I don't do this part. I can look at the histology, but this is where you two hop in. So
I also don't do very much of this. Yeah, so I don't do the 3D imaging, but I do the ultrasound imaging, and I have not been using ImageJ. I'm using QPath now, but basically what you do is you just outline whatever it is that you're looking for in the image, you annotate it, and then you can extract measurements from that. So that's what I've been doing with histology and ultrasound images. So I take an image of each of the quarters, and then that's what I do with that, and with the histology images. Yeah, the 3D.
Oh, yeah, so for the 3D imaging part, it's pretty similar where basically you have like a 2D plane is how I describe it. So you have a piece of paper and that's a normal image, right? It's just on one dimension versus a depth image is making it into a matrice. So it is exponentially increasing the amount of data that you have. So we work with a lot of data. So instead of just looking at, okay, the location, left versus right, up versus down, you know, X, Y axis, now you're working with a matrice worth of data. So feature extraction with that is a little bit more convoluted, but you can get things like volume, area, length, eccentricity, which is like it's like circularity, but a little bit different, without getting too in the weeds with technical terms here. So with this, you can train an algorithm to separate the udder into the 4 different quarters based off of these features. So the depth, looking at how far away is it, plus the location of the different features. And then you can also use a one-dimensional autoencoder. So an autoencoder, putting it very, very simplistically, is basically where you get data and then you train a model to make data that looks like that data but is unique. And that's a way of kind of increasing the robustness of the data that you're working with training an artificial intelligence model. So I am also a nutritionist turned data scientist. As Joe was, I work with really, really great computer scientists who I'm sure could have explained that much better than I just did. But that's kind of how the 3D imaging works in a nutshell and how we extract features from those types of images.
sorry to put you two on the spot, but you two are the ones that analyze the data, so better for you to explain than for me to. So when we were working on this, we had to figure out how much data do you actually need to know it's significant, right? We have these sample sizes, and in genetics, their answer to me is thousands of more cows, more cows, more cows, more cows, and you're like, what are we even searching for now? But we had to figure out how sensitive our technique was. So you're gonna do this ultrasound, how sensitive is it, and how good of a predictive measure is it? So we thought about some different ways we could do this, and what we know about mammary gland biology is that in growing animals, there's 2 different periods where you can affect how the udder or the mammary tissue changes. When they're young animals, you can feed them a high plane of nutrition, so they're getting lots of protein and energy and fat, and then they have a lower plane of nutrition that will change the growth of that mammary tissue, basically just on the sheer amount of protein and fat they have in their diet. So we knew that that was one time period, and then another time period is when they get to be a little bit older, 8, 10 months of age, similar type of thing where they can have this influence of lots of nutrition and lower nutrition and how that might change the composition of their mammary gland. So in order to figure out if this would work or how sensitive our technique was, we did a pilot study that allowed us to like correct things for the bigger study that Alysia did. And yeah.
Is the green light on?
No. Now it might be. Sorry. It's a good thing I'm loud, or at least that's what my husband says anyway. So we had these calves and they were born. We have calves being born a lot at our farm because we have quite a large farm. And we had calves and we paired them. So we were assigning one calf to a higher plane of nutrition and another calf to a lower plane of nutrition. And we matched the amount of like grain besides milk that these calves were getting to their high counterparts, so that the lower ones getting less protein and fat couldn't overeat, right, to make up for that calorie difference, because we wanted to see this difference, because we needed to be able to figure out how good the ultrasound was. So we did that, and we also had to do another thing. We needed to take mammary gland biopsies, because part of this model is also saying, is that like ultrasound image reflective of the tissue that's going to become the milk secreting tissue? And you don't really know unless you look at a section of that tissue and say like, there's the ducts, there's the parenchymal tissue, so that we know we can relate to what's being shown on the ultrasound to what actually is in the udder itself. And that's because an ultrasound will not show you that. It will not show you necessarily what you're looking for. So we had to figure out how to do that, because people don't biopsy young calves. And we worked with the vet staff here. At that point, I'd biopsied a lot of different stages of cows, and I'd convinced them that we were good enough to try this. And so we had really great help from our animal care staff here that supported us and helped us refine this technique. And so we were able to take mammary biopsies, because most of the previous literature, they had to euthanize the animals to take the tissue to really look at it. And we don't want to do that. We wanted a longitudinal study, and we also don't want to euthanize an animal just to take a section of tissue. So we did that. We followed these calves for 9 weeks, and we were analyzing their body weight. We were doing some other measurements. Joao's group was doing some other measurements as well. And then we were taking the ultrasound images and doing biopsies to figure out what this looked like and then what we would actually see. So we took body biometrics, body weights, and we're recording their feed intake as well. So this went on in August of 2020. It was a 9-week study before we started the larger study. So what you can see here on the left is an ultrasound image from the same calf over the 9 weeks. And there's a yellow circle around the black area, and that's the part of the tissue that's going to grow over time to become the lactational tissue. So you can see 1, 2, 3, 4, 5, 6, 7, 8, 9 weeks, it's growing. And we can identify the shapes growing. And then this is the part where when we take the ultrasound video, Alysia is able to like mark around this area, and then she has to segment it so that you can do the measurements on black-white imaging, that you can't extract the features just from the ultrasound video. So she's had to do this on a lot of animals and a lot of ultrasounds. So we were able to sort out that we saw a difference. This sample population was not enough of a representation. We only had 15 per group, but we were at almost significance because of the dietary treatments, and it helped us refine our dietary treatment for the larger study. So we didn't actually have to go as low of a nutritional value as we originally thought. So then this is when the bigger study really took off, and we figured out when we should actually start biopsying the tissue, how we weren't sure how frequently we could, we weren't sure what the tissue would look like. And this here shows you, this is probably a 9-week biopsy, right, Alysia? 10 weeks. So you can see how small that tissue sample is there in the Petri dish. But this is what the histology looks like. So when we do an H&E stain on this tissue that's been paraffin embedded, you can see these dark purple areas where there's those ductal structures. And then what you see here, this is just a representation of the growth of these animals over the first 15 weeks of their lives. So the dark yellow is the body weight high. And then the lighter yellow is the body weight of the lower nutrition calves, and then the dark red is the parenchymal area, or the secretory tissue area from the high, and then the orange is the secretory tissue in the low. So you can see those differences. We created actual differences in our two treatment groups. And we only did the treatment so that we could figure out what the variation was in our sample calculations. This is histological sections from 8-week-old samples. So we figured out we weren't going to do 8 weeks. We went to 10 because we weren't seeing much difference between them. So this was another part of the study, as we were able to figure out the optimal time points in which to do the sampling and what the differences might be. And then we were able to wait a little later in the animals so that we didn't have to do it so young. But you can see that. And Alysia mentioned this program, QPath. She's very, very inquisitive, and she always finds better ways to do things. And she found a new camera for our microscope that allowed us to scan the whole slide. We have an older microscope, but she was like hand stitching these images together. So she'd take a picture here, then one here, then one here, then one here, and collapse the images stitched together. Because in order to train these models, she has to annotate every little duct has to be counted. So it was really important that she found that, and while still lots of time, reduced the time tremendously by which she had to do this work. So everyone is always like, this is a great model, we're gonna have this trained model, it's fantastic, can I have it now? And I'm like, this is the AI right here. So AI is built off of real data, real people, real biologists doing things. It has to be trained. And that's one of the things I would say, and what I've learned is that, and Joao would always say this too, is that you always need a retouching of it back to the real data because it drifts, just like a lot of things do. So she's the original AI of this project. So what you can see here, we had an animal that we had to take off of study due to illness, independent of what we did, but we were able to, we took pictures so that you could see in that circle area is what she's actually ultrasounding, that dark pink tissue. That's parenchymal or lactational potential tissue. And the wider tissue, at least in ruminant species, is largely connective tissue. It's not fat. They have pretty low adipose in their mammary gland compared to humans or rodents. And you can see those two arrows. Those are two, I think those go straight to where the teats would be on the animal, so they're tiny little cisterns, so to speak, because this was quite a young heifer. Well, she was like 6 months? 6 or 7 months when we did that one. So it was an opportunity to really she was able to ultrasound and then dissect this to get good reflection of where we were actually at. So I'm going to let you take over now, because this is where you get to talk about your project and all the fun things.
Yeah, so I followed those animals for 9 facility, and so I'm still kind of following them on Dairy Comp, and these are actually their babies that were born in Arlington, so I thought I'd include that because everyone loves calf pictures. And then these are my calves from my current study. I always like to point out the one on the bottom right. He is a special breed.
And if you want to follow up with her later and come to her office, those are hanging up above her desk.
Yes, and it's been updated, so it has more photos now. But yeah, Laura took this picture of the wall at my desk, and it has pictures of all my heifers from this study. And so for this long-term study, we started out with 210 animals, and we kind of fed them the same as our pilot study, where we had animals on the high and then a pair-fed animal on the low diet. And then we're following them through the first lactation this time, not just for 9 weeks. So the point of this study is to kind of validate the use of ultrasound as a non-invasive approach, so we don't have to cull the animals to take their tissues to look at. And we took weekly blood samples from week 1 through week 8 or 12, week 12. And then when they were about to hit puberty, I was taking their blood samples weekly for about 4 months there until they hit puberty. And then I also take blood samples every time I do a biopsy, which is listed there. So we do the biopsies. I did the biopsies on about 120 animals at 10 weeks of age, which is just a subset of our large group. And then a smaller subset I've been following for a longer period of time. So I've done their biopsy at 6 months, 9 months, and 12 months of age. And then once they went into gestation, I did their biopsies at 5, 7, and 8 months gestation. And then I just actually started doing the first day after calving biopsy, so 1 day in milk. And then during lactation, we're also doing day 45, 90, 150, and 270. And then I just added more biopsies after dry off, so that's new. So I kind of wanted to get the whole lifetime of biopsies, so I'll have a nice collection. So here is pictures from me doing biopsies. So the first image is me doing an ultrasound. So typically, before lactation, what you're doing is you're trying to find the tissue, right? You're trying to find the parenchyma, and that's what we use the ultrasound for. And then once they start lactating, what you're trying to do is, because there's so much parenchyma, you could literally punch anywhere on the udder and you'll get parenchyma, but you're trying to avoid large blood vessels. So in the middle there, I am taking a punch, and Ariana actually took this video last week. So we usually use a core punch. At this age, this was 5 months gestation at Marshfield, and we used a 6 millimeter punch. So at 10 weeks, we used 2 millimeters, which is very tiny. That's why we get such a small amount of tissue. But at this age, we use 6 millimeters. So I just took a punch, and sometimes it takes a while to get the tissue. But there's the tissue, and then I hand it off to Ariana. And typically what I will do is, what we do is we will split the tissue up, so we save some for RNA, and then that has to go into liquid nitrogen. And then the rest of it, we put in formalin, and then the next day I will usually transfer it to 70% ethanol. And that's what we make the histology slides out of. And then the third picture is just me suturing. Have a lot of experience now. And then it's just some examples of fun things that happen during the biopsies. So yes, they pee on me sometimes while I'm suturing. The image on the right, I did a biopsy in Rachel, and she squirted blood all over me. So
You should note that all her cows have names.
Yes.
She knows all of their names.
So here are some examples of tissues that we might get. So the one on the left is from a 10-week-old animal, and it's about 6 millimeters by 2 millimeters. So it's very, very small, and that's with the 2 millimeter punch. And then the images in the middle and to the right are from older animals. So as you can see, as they get older, we can use a larger punch, and you get a lot more tissue. And then here's an example of a 12-month animal. Her name's actually Laura, named after Laura. So this is an example. So this is a histology image on the left, and then on the right is with the annotations that we do in QPath. So if we zoom in on that, this is what it would look like. And so everything outlined in the red is the adipose tissue, and then the purple is the ducts from the parenchyma. And then I also outline the entire tissue, so we have a measurement of that as well in magenta. And an image like this will take, I think this one took about 8 to 10 hours to do. So it takes a long time. So this is my girl Emily at 1 week, 10 weeks, 26 weeks, 39, 52, and 92. So the images at 10 weeks, 26, 39, and 52 are at the same time points where I did her biopsy. And then at 92 weeks, that's when she was about 8 months pregnant. And then 95 weeks was a few days before she gave birth. And then that last picture there is her baby at 2 days old. So these are Emily's images from when she was at Arlington as a calf. So because of the pilot study, we figured out that we don't actually have to take images so frequently, because for the pilot, I was taking images twice weekly for 9 weeks. Whereas for this trial, I actually took images once every other week, and that worked out really well. So as you can see outlined in white, that's the parenchymal tissue that Laura had spoken about, and it does grow over time, which we can see in the ultrasound. And then here's some videos of Emily at older ages. So at 6 months, 9 months, and 12 months, which is the biopsy time points. And as you can see, the tissue changes a lot over time. So if you look at 11 weeks there, it's kind of just like, if you were to feel it, it kind of feels like an almond. It's kind of like a hard mass of tissue, whereas when they get older, it kind of spreads out, and you can see the ductal growth upward. And then I also take images further on during the biopsy. So this is during gestation at 5, 7, and 8 months, and then also her 1 day in milk biopsy. So I'll play those videos for you. So what's really cool is at this point, you can start to see like a little cistern in there, and you can still see the ducts, but at this point, it's so fine that as you go higher up in the udder, you don't really see it anymore. And then the really cool one's the one where she's 1 day in milk. So what I typically do is I'll start at the teat, and then I'll go all the way up the udder, and that's how I get my images. And that one was actually so long I had to stitch 3 videos together. So here is from my first publication that just came out. This is Emily again, but so the top row, they're all images from 10 weeks, 6 months, 9 months, and 12 months, and that's more near the teat, where you can see a lot of the parenchymal tissue, and then the larger ducts. And then the second row, that's approximately where I did the biopsy. So what we're trying to do is stay far from the teat so you don't damage the cistern, but at the same time, you're trying to get tissue. So that's what we use to locate it, the ultrasound. And then the bottom row is just images, the histology images from the same time points. So as you can see, there's a lot of change over time. Typically at 10 weeks, we don't get too much tissue, so there's not a good representation of what the tissue really looks like, but as you move on older in age, there's a lot more tissue, and you can see that there's a lot more adipose tissue in the histology images, which kind of correlates with what you see in the ultrasound image.
Okay, so I'll kind of transition here and go into the 3D imaging, and then I'll talk some more about data related to the lactation biology, of course. So this is a video, I believe. Yeah. So this is a video kind of that 3D image pulled up in a software where we can look at it, manipulate it, and it's where we get like a 3D point cloud, it's called. So all of the different points on the mammary gland, as you can see there, the 4 teats, and then the 2 legs off to the side. And if you see bigger things like the green and the blue over there, that's actually a person that's kind of like peering down with the camera. So you can have a pretty good representation, and also with this, you can measure the distances between the teats, the distances between the quarters. It does a really good job, the 3D camera does. So that's a way where we can kind of take a depth image. So the point kind of transitioning here is it's really, really cool what Alysia and Laura are doing with ultrasounding and histology. We're trying to see, okay, can we kind of take it a step removed and just using 3D imaging also predict maybe lactation potential and other factors there. Yeah.
So these are my animals that have recently become cows. So they all just started calving in mid-April. And so now I have, these are 10 of my biopsy animals. And then there's also 3 who are not biopsy animals who recently calved. So lots of calves. It's calving season. And since ultrasound is so cool, I thought I'd include some images of my animals' babies. So I had an internship where I was learning more about ultrasound, and so I learned how to do repro ultrasounds. So I was practicing on my animals, so I was taking their images random times during gestation. I thought this was really cool. So the first image is at 48 days, and then the second image is at 84 days gestation. And it's incredible how different they look, just 40 days difference. And then that's Emily's baby once it was born. And here's just some other ones that I thought were really cool. So it's 41 days, 56 days, and 63 days. And then these are their babies that haven't recently been born.
So kind of going back into more of the 3D imaging work. So our part of the project that Joao is collaborating with Laura and Alysia on. Thank you, sorry about that. Okay, it's green now. So from the same study with the 200 calves that are now heifers turning into cows, first lactation cows, we collected images at least weekly, if not every single day, from these animals since they were born. We collected infrared and depth images, and we have also a camera system installed at the farm with about 40 cameras that are able to collect images, and I will talk a little bit more about that later. So we want to see, first of all, individual identification of these animals, because the goal is to have kind of an automated system that can track animals on a farm and collect images completely non-invasively without intruding, without even having to go in the pen with them, and taking 3D images. And from that, we're hoping to see, is there a correlation between body shape, so just the features and their rump and their length and their height, is there a correlation between that and their first lactation potential? Without looking at the mammary gland, without looking at the histology, is there a way to roughly predict how well they will perform? And also, is there a way to see, what kind of hormone levels they have? As Alysia said, she's been very diligently taking blood samples very often, so we have all this data. So we're hoping maybe in collaboration, we can just look at body shape changes and predict things like estrogen levels, progesterone levels, and kind of give an insight on farm of what's happening to these animals without maybe even having to collect data from them other than these images. So my job is a lot easier than what Alysia does. I just have to kind of stand behind the cows with a pole. In this video you can see. And then on the right is the correlating depth images that we're getting from those animals. So again, I have a pretty cozy job. It's really not that difficult collecting the data. And this is done in the pens where there isn't a camera system already set up that I'll show in later slides. But then you can see that depth image, and you can also see me. It's a little bit small here on the screen, but looking at the distance between the hip bones on that animal, kind of looking at rump shape with that depth image. And then where I don't even have to do anything other than replace USB cables and keep cameras from freezing sometimes in Wisconsin winters, and just general maintenance, we have a camera system set up at our Marshfield facility, and we're actually working on setting up some in our Arlington facility right now. And you can't really see it super well, but on top of each of those pins, on one of those poles, there's kind of like a newer looking wooden slat going across, and there's an electrical box set up there. So each of those electrical boxes, which in the picture on the right is highlighted by an orange box, contains a small computer called a Jetson. And then on the picture on the right, the yellow boxes are highlighting the cameras that are set up over the water in each pen, because it's guaranteed that each animal is going to go to the water at least once a day, if not a lot more. And so that's a way where we can collect that depth image. So the computer that's kind of set up there next to the cameras has a code written on it to collect an image anytime the depth is above a certain threshold registered on those cameras. And it collects an image, and then it sends it to the cloud or to an external hard drive, depending on where it's at in the barn and how good the Wi-Fi signal is. Also, something we're working on is Wi-Fi on farms. But we've got a good system, and as I highlighted on the first slide, I have about 70 terabytes worth of data from this. So a lot of data. I like to say a picture speaks 1,000 words is the cliche I always use. And it also involves a lot of data that you can store. So the whole point of this is that, okay, how can we take this kind of data that we're able to get, the more physiological data, the lactation potential information from all the biopsies and blood samples, and how can we use that to also train a model based off of body shape to see how well these animals will perform, and hopefully we can apply this to industry dairy farms. So in this example, we have cameras set up on the exit lane of the milking parlor. On industry farms, cows get milked twice a day, if not 3 times a day. If we have a camera set up there, and it's guaranteed at least 2 or 3 times a day, they will have an image taken of them. So then we can track their body shape, their changes over time, and maybe, possibly, the hope is, predict how well they will lactate. And also, when they're lactating, maybe how their mammary involution will change, their dry off, things like fertility. Really, there's a lot of information that we can get from images, and we try to find out all the different correlations we can make there.
And it's back. So yeah, our future goals and what's neat about being in a department like mine is that we have a really robust set of scientists that all do very different things, which I don't think a lot of people realize about an animal and dairy sciences department, that you go from biomedical to data science to geneticists to nutritionists to immunologists all in one, like, well, it's not little, the very tall, awesome looking building over there by the volleyball courts. I said awesome, see? But there's a lot of different people doing a lot of different things, and we're really able to synergize a lot better than what people, I think, realize our capabilities are. And just a conversation one day led to a really cool project that has implications not only for animal agriculture, but also for human medicine that no one would have realized. And the fact that you would have told me when I first started here in 2011 that we'd be using like the same kind of information that my iPhone does to unlock my screen from my face, never would have suspected that. So our future goals are to develop this model and then use it in practice. On farms, of course, this will be a nice thing that people can use as their growing stock are there as better predictive models. And it's non-invasive and much easier to do than things that we do currently. I'd also say about this data set is that surprisingly, there is not one that exists where people have longitudinally taken samples from that many calves or cows starting at life through that period of time and taken histological sections that even in vet journals, much to our surprise. So that in of itself, from an animal health perspective, is going to be really nice for looking at that sort of thing. But we can also utilize this model in practice in humans. And there's a family medicine doctor that's coming in August who is really interested in our work that Joao and I have met with. and she's going to be doing some research track work here as well. And she knows the colleague I did my sabbatical with, and so we're hoping, and she's hoping that she'll be able to at least start to pilot this in clinic, where we could use the ultrasound in humans. So that would be my dream. I saw one too many women crying in an office or at the hospital because they couldn't make enough milk and they didn't understand why. And so I think that this will be a potentially really nice way to help women develop a lactation plan just like they would as a birth plan. And then utilizing these very sophisticated machine learning methods to predict lactation capacity, of which Alysia and Ariana are doing a lot of the training work. I think sometimes when we say we want to use these techniques, people don't realize that it takes an original human and an original biological data set to train these models. And so it's an enormous undertaking, enormous. We have a geneticist who works with us, and one time he told Alysia, "Why is this taking so long? Why do you need more help on the farm?" And we were like, you know that flash drive or the Jetson pack where all the data comes out of? Alysia collected all the data. She took care of all the animals. And so it really is a myriad of expertise coming together to use our strengths to have these collaborative projects, which is where I see science continuing to grow. We're not in isolation anymore. And so I'd really like to be able to personally, it's been my passion project to have this be a way that you can help counsel women before giving birth in terms of what their lactation potential might be. And really dreaming big, I would more than anything in the world love a Gates Foundation to be able to go do this in areas of the world where we could potentially utilize this technology to help support women in different countries where they don't really know what it's going to look like, and they particularly don't have access to clean water, so formula is not a good secondary option. So those are the big life goals. I dream big. So on the left is my lab, which is made up of a lot of physiologists, and on the right is the digital livestock lab, which is Ariana's PI's lab, Joao Dorea, and my daughter defected. So you see the very tiny little girl on the digital livestock lab? That's my daughter. So she likes computers. She does love animals, but I think she prefers using a computer than working for 10 or 12 hours a day out at the farm. So I lost this battle. But nonetheless, we do a lot of fun things together, and Joao's done a really great job doing stuff at the WID, where they often have a setup with the calves, and they're showing all their really, really sophisticated work they're doing, and it's been a real pleasure to work with him. And of course, really work with them because they really are the bosses of us. So as much as Alysia likes to say that I am the boss, she is. So nonetheless, thank you.
I understand how taking pictures of the animals from above and measuring things like their hip bone structure might relate to gestational or delivery issues, but how does that relate to lactation potential?
Yeah, so that's a really good question. Something that I have come to learn going into from things that we can't. So there has been previous research showing that the rough shape, so yeah, the hip bone difference and kind of the overall shape of it,
So I have a general question about computer science education among yourselves, and remind your daughter that it was Count Lovelace, not Charles Babbage in 1852, who did the programming. And the new vision, to my understanding of the University of Wisconsin Computer Science Department, is to have everyone at the University of Wisconsin be familiar with computer science. So I wonder what the three of you understanding is, and the basic concept of garbage in, garbage out. And we're all challenged by artificial intelligence, and I think you've presented us with a wonderful look at what we can do to move in that direction.
My foray into computer science was like a very ill-fated time working with SAS, and now I'm really glad for the people that are in our basement that know how to use SAS computing. I'm much better in theory, but I do think, and this is a discussion that Joao and I have a lot, is this too much of a reliance on the computer science without going back to check the actual living, breathing thing can lead you in the wrong direction if you haven't gone back every once in a while to like validate your data set, to make sure that the artificial intelligence or the machine learning is actually still trained to the correct model. I think of it as when you like breed animals for a long time in our mouse facility, 18, 20 generations, the C57 Black 6 that I bought from Jack's isn't quite the same as the C57 Black 6 at Jack's. It has some genetic drift. And so I really see that we can continue to have those things together, and that we can build these models and they can be more sophisticated, and understanding this stuff is so important. I'm amazed about what they teach in the elementary school learning at this point. My kindergartner is learning how to code. I can still barely use PowerPoint very well, and I'm not that old, but I'm old enough. And so I really think doing that stuff together, but not losing track of biology, and that we're all individual animals that have variability. And so I think that's sometimes what I feel that computer science might miss occasionally, is the extent of the variability between, one individual to the other. And so that's what I find really fascinating about working with someone like Joao, who always goes back to that sort of scenario. He doesn't just rely on, a data set that has been built for the 100 years or whatever.
Yeah, I'm not sure if I have too much else to add to that. My background is an animal nutritionist. I did my master's in ruminant nutrition. I had not taken a single computer science class before starting my PhD here. So Joao really values, as Laura highlighted, balancing computer science with animal scientists. So it seems like for each computer scientist he adds a group, he adds an animal scientist. So that's kind of how I came in. But since my PhD, I have taken nothing but computer science classes. So I think there's a lot of value, especially concerning research and how far we can go and how much we can learn with an understanding of computer science. But again, like you highlighted, garbage in, garbage out, you need to know what data you're collecting, the experimental protocol. You have to have an understanding of the biology. So I think as an animal scientist by training and background, I think I'm able to bring some insight into data that some of the computer scientists can't, and vice versa. They can also find some ways to analyze the data that I wouldn't have thought of. So it's really a balancing act, but I think there's a lot of promise for the future.
This is a quick question. Actually, a lot of this. Oh,
Oh.
The human computer.
I don't do much with AI, to be honest, but I think it'll be really helpful, especially for tasks like annotating an image, where for that one paper that I just published, we annotated well, mostly me, annotated about 72,000 objects. So it takes a lot of time. Like one image can take me 8 to 10 hours if it's very complicated. So using something like AI can be very helpful in research, but like they said, it's also a balancing act, and you have to know what you're doing and what you're looking for. So it can be very helpful.
So, I have one biology question and one computer science question. I'm not a biologist, I'm not a computer scientist, I'm a clinician. But the biology question is that not being able to lactate, it's gotta be a very strong tendency not to survive in an evolutionary time scale. So the fact that 5 to 15% has trouble is shocking to me. It's gotta be a modern phenomena where people don't always have to run around the savannah to survive. So the things like, obesity, nutrition, birth control pills, because I don't see any less than developed countries having lower birth rate or even breastfeeding rate. So that kind of makes sense to me. It's puzzling to me. Yeah.
That's a great point, and certainly that is part of it. The other thing too that's different than like the great apes and some of the monkey species that predate us, they stay with their young like 5 years. I think gorillas, it's like 5 years. Their inner birth interval is much longer. And so part of it is related to like they breastfeed for a long period of time, and that somewhat suppresses you from getting pregnant again, and it decreases that inner birth interval. And then there was a period in time in which that inner birth interval started to decrease, but also some of the apes and monkeys, their offspring have to have milk longer for survival than humans do. So humans are okay like on milk only for the first 6 months of life. And so you're able to switch them. But obesity, nutrition, and then there's a whole other area of very sexualized context surrounding breastfeeding, where like in France, they don't like to breastfeed because it's very like sexual in thought, and the United States is a lot more like that. And then in other countries where they'll feed their baby or their small child till 5 years of age, and there's not a sexual connotation to it. And then there's also been these switches with socioeconomic status, where like the period of time when the rich people had all the wet nurses, so all of the poor people were providing the breast milk to the babies, including their own and whoever they worked for. And then now that's since flopped, where more of, at least in the United States, the higher income individuals are breastfeeding more frequently and longer than the lower socioeconomic status individuals. And so there's a lot of like disease related to nutrition, obesity, et cetera, plus those sorts of things. So
it's probably not genetic. I mean, that would be more pattern recognizable. Now,
Right.
The computer science thing, I'm not a computer scientist. My knowledge is from YouTube videos. But when you're modeling this, are you doing it as a regression model or is that classification model? Is that lactators versus non-lactators, or is this a mount?
Yeah, so that's a good question. I think initially we always go for classification because that's an easier way to look at something
get
if there
good
is
numbers.
potential. Yeah, it's just is it so I have a project where I looked at the body shape images and pre-weaning. So in the first month of life of those calves, and then I used that to predict when they were going to start cycling. So from that initially, I just looked at it as a binary. Were they going to start cycling, I believe, at 8 months? Does that sound right? Okay, 8 months. I have to check with Alysia. She knows everything about these animals. If they were going to be cycling at 8 months or not. So it was a binary classification, yes or no. With that, we saw really good results. So the next step is to look at it as a regression of what were their actual like estrogen and progesterone levels around that time.
So
So both.
Yeah, I'm asking that question because if you're going to use it in a clinical setting, you may wind up having to develop both to answer questions that people practically do ask.
Yeah, absolutely.
That's a really great point. And one of the things, like when I given this talk to some nurses and breastfeeding medicine doctors, and it's really interesting because we do take blood samples and we're looking at progesterone and insulin and some other hormones that we know to be associative with poor development of the mammary tissue or better development of the mammary tissue. And one of the things that I wish we could do, but of course, like I am the mother to an 11-year-old daughter, and like you would never convince a parent to say like, let me take the obesity measurement of your child and their hormone structure and then figure out like, would there be an issue later in life? I suspect yes, because most obese women have a delay in the onset of lactation. If they do make it to the point where they choose to keep breastfeeding, which that demarcation is 72 hours postpartum, they often come back and they make more. But that first 3 days of actually setting the stage, they struggle with. And there's a bunch of other comorbidities like hypertension, et cetera. And so there's a lot of value we have from some of the hormones too, which with this project, having like the tissue development, the hormone analysis, the ultrasound, it's going to be a really robust data set that allows us to do some of the binomial data plus some of the actual, different variations on specific hormones, because that would be exactly something someone would ask. It's a great point.
Actually, can we do this for now?
Oh, okay. He's coming to you next, he says. He says.
Yeah. So I'm a biologist that has a lot of data friends, and I agree that we probably could look at like physiological data to make predictions. I want to do it in blood because even clinical blood samples have lots of data that we don't really look at and could correlate with a lot of the stuff you're doing. The body structure might be a little bit different simply because of some of the information Dareth brought up. I would like to, since I've considered myself an educator, one of the books you should look at is Biochemical Individuality that was published in 1956 that is still on Amazon. And it describes exactly what you guys are working on. Laura, you made the right points that there's a lot of variability, and it's going to be very hard to say that variability in structure of the bone structure or the rump structure is going to have something to correlate with fertility or milk production, simply because of the variability in all animals. So it's just an add-on to what you've already discussed, really.
That's why we thought it was so critical to do this type of work, because we have like real samples with real measurements, and then we have the image analysis so that everything's not just correlated, it's like real potential cause and effect. And one of the best things that I think has happened in data as of the last few years is showing the dot plots. Like instead of standard errors or standard deviations around a mean, because it's great, the mean looks nice, but like you might have 10 up here and 1 down here, and what are they doing? And so I think that's what's really nice about this. Yes.
Yeah, and a really cool thing about the artificial intelligence and machine learning is that it can pick out the similarities within the noise, right? So there's a lot of individual variation, but it can also pick out what's similar between all of these related to this one factor versus all the individual variability, all that noise in it. That would be very hard for us to pick out because of the way that the data is manipulated in order to train that model. Yeah. Mm-hmm. Yeah.
My question is, with regard to picking out, taking 10 hours or so to look at a picture and then highlight areas, what are you highlighting and why are you highlighting them? I think you suggested that AI might help you with that. So as part of training data, highlighting certain sections of your images, why are you doing that? And for what purpose?
Yeah, so because we have histology samples from different ages, what we're looking at is development over time and comparing that over time. So what you're looking at here is like the ductal structures, and if you, let me find the slide. So for this image, as you can see, there's a lot of change over time. So in the first image at 10 weeks, we don't typically see a lot of adipose tissue in the histology images, but then I think a little past like 12, 13 weeks of age is when you start to see the mammary gland parenchyma like going into the fat pad, and then you start to see more adipose tissue in the histology images. So by annotating the images, you can quantify how much tissue you have and what kinds of tissues that you have. And you can even use different stainings like trichrome staining to differentiate between different tissues. And so that's what I've been doing. And so what we're doing is following the development over time. And so that's why we annotate. And then using AI would just make it a lot faster.
Yeah, that's what I was thinking, that AI would be able to pick out
Right, yes, but
those images, and you're not there yet.
no, we're not there yet.
She's doing the training first.
Yes.
And then,
Yeah.
Yeah, up to this point, I've only annotated up to 12 months of age for all my animals, and that's about maybe 140 images. So we still have the rest of their lifetime to annotate, so there's plenty of images to annotate.
I'll be right up there, Sandy.
Well, I had to look up that cows don't live long enough to get breast cancer after the news of artificial intelligence and breast cancer. But I wonder, are there any genes that correlate between cows and humans that you're looking at?
That's a great question. They don't generally live long enough to get breast cancer. The structure of their tissue also gives them some prevention. I hate to use prevention, but it protects them from cancer because they don't have as adipose tissue as like a human breast or a mouse or a canine. For all species do get them. But it depends on what you're looking at. So another area that I spend a lot of time in, I study peripartal animals, humans, cows, rodents. So I study phenomenon related to metabolic problems. One of them is hypocalcemia around the time of parturition, how that exacerbates like different metabolic things that the animals do to like, make milk and stay healthy and provide enough nutrition for their offspring. And so we have started to see a few genes that might be conserved. I'm working with another geneticist in our department, and he has picked up some genes around maybe a calcium transporter and a vitamin D related family protein that might be conserved that could be related to that. Mammary gland actually has one of the lactating mammary gland has one of the most conserved genomes because they're all making milk. So a lot of lined up all the genes expressed in a Tamar wallaby, a platypus, a human, a cow. The milk proteins are all, they go up, and it's like a percentage magnitude that is pretty similar across species. So there's quite a bit that's conserved across in terms of making milk. Some of these things related to disease, whether it's cancer or metabolic or immune function, some of those are a little more nuanced. It just depends on what your research question is. But
Any other questions? If not, thank you very much. That was fantastic.
thank you.
And then, as you know, we adjourn to the library afterwards. And then next week is Kelly Senecal and More Than the Tailpipe, looking at the future of internal combustion engines. Hope to see you next week.